So I'm not going to be talking about women in STEM. I'm going to be talking about multiple sequence alignment. And this is somewhat similar to the tutorial I gave last year, I think, but it has some additional material. So some of it should be new to most of you. So multiple sequence alignment is a problem where you have a collection of sequences and you're basically pulling them apart, stretching them out so that they line up. And when they line up, everything that's in the same position has a common ancestor. That's the basic idea of a multiple sequence alignment. Almost every problem that has been posed is NP-hard. It's really foundational for many downstream analyses. And because of its impact, it's been identified as one of the most important problems besides phylogenetic estimation. Well, anyway, the National Academy said that it was one of the most important problems in bioinformatics, period. So that was a while ago. But MSAs, multiple sequence alignments, are used in phylogeny estimation. They're used in predicting protein structure. They're used in annotating genomes. And they have many other applications. And the one that I work on the most is phylogenetic estimation. So you have this pipeline where you start by deciding what you want, what species you want, and which genes you want. And then you're going and getting your data. And then you're computing multiple sequence alignments on each of the genes. And then you're using that to get gene trees. And then you're using the alignments and/or the gene trees to get a species tree or a phylogenetic network. And then you're doing things with that phylogeny, whether it's a tree or a network, to estimate things like branching lengths and times, and do things with biology, make discoveries in biology. The alignment step is so early in this that it impacts everything downstream. So if you have problems with your alignment, everything afterwards could have a problem. And that's one of the things that makes it particularly important. So insertions and deletions are the main mechanism that create the problem that requires a multiple sequence alignment. So you have sequences that are of different length because you have insertions and deletions. But if you know the insertion and deletion history, you can write down the correct pairwise alignment. So for example, here, this collection GGTG was deleted. So there's nothing in the bottom string that corresponds to it. And similarly, this T was changed into a C. Now, you could also say this A is the same as this A, this C is the same as this C. This is called homology. So homology has to do with this relationship. So you can get the true pairwise alignment, which is the homologies. And from that, in a tree, you could get the pairwise alignments on the edges of the tree, and by transitivity, you can get the true multiple sequence alignment. So 3 sequences related by evolution, you can write down all of the homologies, get the true alignment. So true multiple sequence alignment in this context has to do with evolutionary relationship. So how do we compute these things given sequences where we don't know what happened? How do we try to estimate that true alignment? There's a lot of variations on the problem, but one is pairwise, another one is multiple, local versus global. And a lot of the cases where you're just talking about pairwise alignment and you're aligning the full sequence here to the full sequence there, that's a typical like an edit distance problem. It's polynomial time for 2 sequences. And we've all seen algorithms for this. Needleman-Wunsch is an example. If you do local alignment, then you have Smith-Waterman. Everything is straightforward. 2 sequences, straightforward. When you do multiple sequence alignment, you actually have to think about how do you extend from 2 to more than 2. There's lots of variance there. Some of them are more sensible than others. A lot of these problems become NP-hard. And so you end up having approaches that are essentially heuristic. Now, you'll see this thing that says Bayesian estimation. Instead of solving an optimization problem, you want to do a statistical model, and you want to sample from what you're getting from under that statistical model. So standard approaches for multiple sequence alignment. One way to do it is you take one sequence of your collection, and you do pairwise alignment to that one sequence. And because you've done pairwise alignment to everything to that one sequence, you can infer the multiple sequence alignment. So that's called star alignment, and it's actually pretty common if you're doing genome alignment. So there's one genome, you do pairwise alignment to that one genome, you're done. With sequence alignment, it's not necessarily as common. And it's not that bad if everything is closely related, but otherwise it's just not that reliable. Progressive alignment is something where I'm going to show you a picture about it in a minute. It's very, very common. The idea is you get your sequences, you get an initial alignment, and you get a tree based upon that initial alignment, and then you realign using the tree. And I'll show you a picture about that in a second. Then there are things called consistency. You're looking at lots of different alignments, and you're saying, is this homology, this letter to this letter, supported by lots of other letters, X and Y supported by their relationships through Z? So that consistency thing is a way of extracting what you have confidence in. And then, of course, there's machine learning approaches. But back to progressive alignment, it's this kind of thing. You have an input set of sequences. You get pairwise distances in this case. The pairwise distances could be just, the pairwise, the best multiples pairwise alignment you can get, and then you just compute the edit distance. From that pairwise distances, you get a tree. From the tree, you now put the sequences at the leaves of the tree based upon the pairwise distances, and you merge the pairs into an alignment. So like this pair of sequences, this is the pairwise alignment. This pair of sequences, this is the pairwise alignment. Now this and this sequence, you get this three-way alignment. So you have a pairwise alignment being aligned to another sequence, you get a three-way alignment. And you just go up the tree until at the root you have a multiple sequence alignment. Okay? Now, how you do it, how you get the distances, how you get the tree, and how you merge, that's where the variants occur. Okay. How do they perform in practice? You don't always know the true alignment. So again, just like in phylogeny, you can do simulations. So you have a simulation, you have a model tree with a model sequence alignment, which means on that model tree, you evolve sequences, you know the true alignment. So there's their true tree and the true alignment, and you'll notice it's unrooted because even though the model tree is rooted, in the end we're going to be comparing estimated trees to the true tree, which will not be rooted, estimated alignments to the true alignment. So you have this true tree and true alignment, and you take the sequences and you eliminate all the gaps. So now you have unaligned sequences. And then you take that alignment, the unaligned sequences, and you align them, and you get a tree. Now, typically, that tree will be something you compute on the alignment, but you might have co-estimated them together, okay? The point is, you start with sequences that come from a simulation, you know the true alignment, you know the true tree, and you try to construct the alignment and the tree. And now you can compare them, and you can quantify the error. So when you compare an estimated tree to the true tree, you can quantify the error. The estimated alignment to the true alignment, you can quantify the error. And this allows you to explore what happens under different model conditions. Slow rates of evolution, high rates of evolution, lots of indels, fewer indels, large trees, small trees, whatever. So many years ago, I did a paper where we were exploring if we always use maximum likelihood for tree estimation, there's lots of ways of doing trees, but if we always did maximum likelihood for tree estimation, and we looked at what happened when you do the alignment, how much does the alignment error impact the tree? That was our basic question. And we looked at large data sets with at least 1,000 sequences. And this is something that we published in 2009. Every model condition here has 1,000 sequences in the tree. And they range from low rates of evolution to high rates of evolution, more or less from left to right. But they all have insertions and deletions. The insertions and deletions vary in their lengths. Some are short, some are long. And so the top thing is tree error, and the bottom thing is alignment error. So this black curve is what you get on the true alignment. So if you didn't have any alignment error, you wouldn't get too much tree error. So you start off with the easy models, you have about 5% error. The error here is the fraction of the tree that you're failing to recover. So you can get 95% of the edges right, 5% are wrong, you have 5% error. But then with the higher rates of evolution, the error, even on the true alignment, goes up to 10%. Now you're only getting 90% of the edges instead of you got 95% before. But if you have to estimate the alignment, now you're getting a lot of error. Okay? You had alignment error in here, but it didn't impact the tree. Over here you have alignment error, and it's impacting the tree. And the worst one is here, which is CLUSTAL W. So CLUSTAL is like a family of methods. It's been developed over the years. It's extremely well cited. The most recent thing is CLUSTAL Omega. This is back in 2009, so it was CLUSTAL W. You can see that alignment error is impacting the tree. And if you look here, you can see the alignment error. Okay, so high rates of evolution affects the tree error. It also affects the alignment error. But there are some large data sets that are easy because they have relatively low heterogeneity. The overall diameter in that tree is low in terms of the number of substitutions. Okay, so another issue is not just rate of evolution, but the sequence length, heterogeneity in the sequence length. So I was working with Jim Liebensmack on plant transcriptomes, and he said, we really need a good alignment method for like 100,000 sequences. I said, okay, let me work on that. And I worked on it, and I thought that I had a really great solution, and then I found out that I had a really bad solution. I didn't have a good solution. And the reason is that the data set had lots and lots and lots of short sequences. So it was just this really massive data set that was really hard to align. And I had to come up with new techniques, okay? So that's another issue, sequence length heterogeneity. So what is it that makes for an easy alignment? Not too many sequences, all the same length, not very much evolution, okay? That's easy. But large data sets can be hard. They're not all hard. They can be hard. And if you have sequence length heterogeneity, you always have an issue. Okay, so easy versus not easy. So the two things that I've been focusing on are large-scale multiple sequence alignment, where I'm concerned with high evolutionary rates, and I'm concerned with sequence length heterogeneity. But I'm not working on the problem of very long sequences. That's a different problem, especially when you're thinking of genome alignment. You know, you have let's say 100 genomes, and you want a multiple genome alignment. I don't work on that. It's an important problem. It's just not one that I work on. So what I'm going to do now is take you through 5 parts. The first part is how we designed methods to be able to go to large data sets, large numbers of sequences, when you don't have sequence length heterogeneity. So that's a divide and conquer strategy. And then I said, how do we deal with sequence length heterogeneity? And in particular, we're doing that by taking the sequences, and if we can get a good alignment on some of the sequences, we can add sequences into it. Is your hand up because you have a question?
Yeah. Sorry, this is a naive question, but I was wondering, when you talk about sequence heterogeneity, do you mean that these methods that don't perform well with sequence heterogeneity are considered to, like, they're super conservative to a very specific length, that it can't be even, like, you have sequences that are a couple of base pairs different?
No. I'm talking about the, when I get to it, you'll see what I mean. Okay. So part 2 is about dealing with sequence length heterogeneity, where some of the sequences, let's say, are 100 base pairs, but the full length is 1,000. It's that kind of extreme, okay? When you have that kind of an extreme, you need a different technique. The third one is statistical alignment. So Balify, in fact, it's from UCLA. Balify is like the leading Bayesian method for co-estimating alignments in trees, and it's great, but it has lots of challenges, and I'm going to show you some of the work we did on this. And then the fourth thing is this really intriguing problem about the differences between biological benchmarks and simulated benchmarks. And when I get to that, basically I'm going to say I don't know the answer, and it will be interesting to see what you guys think. Okay, so first divide and conquer. So you have a large data set, but you remember that some large data sets are easy. What makes it easy is not too much evolution. So you have a data set which has pairwise distances can be very, very large. If you can make every subset smaller in diameter, you can get good alignments. That's the key idea. So at a high level, what it is you get an initial tree. You get an initial alignment, you get an initial tree, and even if that tree isn't great, if you break it into subsets, those subsets can be much less diameter. And if they have smaller diameters, you can get good alignments. So you break it into subsets, you get alignments on the subsets, and then you merge the alignments. And the typical way of merging alignment is just go bottom up, just like in a progressive alignment, okay? And now you have an alignment on the full data set, and you get a new tree. And you can repeat the process, because as you get a new tree, it'll be a bit better than the previous one. The subset alignments will be better. The merged alignment will be better. The new tree will be better. You can iterate. Okay, so it's divide and conquer plus iteration. There's a class of methods like this. It began with SATE, which was from 2009. Then it had SATE2, then it had PASTA, and now it has MAGUS. This is a sequence of method development. They're all in the same basic category of take a data set, divide into subsets, get alignments on the subsets, merge the alignments. How they divide changes, how they merge changes. And those changes end up impacting runtime and accuracy. So back to this example from 2009, we developed SATE, and SATE is this thing in purple, and this 24-hour analysis of SATE, better trees, which you can see here. It's almost as good as what you get on the true alignment, and the best alignments. Now that was very satisfying, but the gap between what you can get in the true alignment, what you get with SATE, is something we wanted to address. So we redesigned it. And we got SATE2, which could go up to about 50,000 sequences, where the first SATE could only do about 8,000. Then we did PASTA, which could go to 1 million. And then most recently, we have MAGUS. What's really interesting here is that the changes that we did in each of these, from SATE1 to SATE2, from SATE2 to PASTA, we were just trying to increase scalability to larger data sets. But the change that we did in the algorithm design improved accuracy. So it was really funny. We weren't aiming for accuracy. We were just aiming for scalability. And we got better accuracy, which gives insights. The MAGUS thing was not. That was a different thing, which I'll get to. So SATE2 versus PASTA versus MAGUS, they actually have the same decomposition technique, which is different from SATE1. They're dividing into small subsets, and each subset is of a maximum size. And then SATE2 is using the guide tree to merge them up. PASTA is taking the same subsets, but it's like aligning pairs of them. So if you have subsets of size 100, now you're making subset alignments of size 200. But the 200 size subsets overlap. And because they overlap with each other, you can get transitivity and get an alignment on the full data set. And so that transitivity merge was the way that we dealt with how do we merge differently to improve the running time and scalability, and it improved accuracy. So PASTA became the thing that like a lot of people know about and now use. MAGUS was different from PASTA in that it divided into the same subsets, but then it takes these subset alignments and it does something really bizarre. So what does it do? It has the subset alignments here. Remember, these are like you take the tree, you decompose into subsets. Each of these subsets, you get an alignment on it. Those are your constraint subsets, your constraint alignments. When you merge them, you're not going to change them. But what was done was my student said, I'm going to take a few sequences from each of the subsets and use an estimated alignment on those subsets to help merge the constraint alignments. And so now you have this big alignment graph where the nodes represent columns in the constraint alignments. And you have edges between the columns if there's one of these backbone alignments that connects them. So those edges are like guiding you about what should stay together. And now you run this through a clustering method. Which clustering method? It turns out that Markov clustering works really well and nothing else. It's strange. This alignment graph, you cluster. When you get a clustering, this cluster may or may not be legal. What you want is to have at most one vertex of each color in each cluster, because you want them to actually give you the alignment at the end. So you have to be able to put these things together. So you then have to modify this clustering. So there's a clustering phase, and then there's a cleanup phase. What's amazing is how good it is. And I don't really understand why Markov clustering is great and other things are not great. But This was the intuition of my former student, Vlad Smirnov. It was just a brilliant intuition. This is comparison to pasta, and then two versions of MAGUS, one faster, one slower. And you can see pasta error, this is alignment error, is higher in general than the MAGUS ones, especially for the high rates of evolution. So this way of just taking all these subset alignments and merging them through this bizarre technique is great. Protein benchmarks, this is no longer simulated. This is protein. This is from HumFam. These are a bunch of different methods here. This is muscle, clustol omega, MAFT, UP, which is a method I'll tell you about in a bit. Here's pasta, and here's the MAGUS methods. So MAGUS is just doing well on simulated data and on biological data. Okay? The summary for this thing, divide and conquer is a general technique. We're showing it with subsets where the alignments on the subsets are computed with MAFT. But we've also explored using other things for the subset alignments. It's a general technique for improving the scalability of an alignment method to larger data sets. Okay, and it's related to something that John Kesecioglu talked about 20 years ago or something called the maximum weight trace. So this actually goes back in time to something else, which is nice. Okay, part 2, a different technique. When your data set has sequence length heterogeneity, imagine you've divided your data set into 2 sets. One is the full length sequences that you think you can trust to get a good alignment on it, and everything else is the short stuff that you want to add. So you have these 2 sets. You have an alignment on the full length, and now you want to add the sequences from the other set. So this, again, was motivated by this problem. Here's the histogram of the sequence lengths. So the full length is around here. Look at how many really short sequences there are. And then there's some long ones and some extremely long ones. So this was just a really bizarre data set. Once we realized that we were dealing with this, we had to come up with a new method. So the 2-phase technique is the solution that we're using. You take the data set, you divide it into 2, the full length and everything else. You get a good alignment on the full length sequences, and you add everything else in. But how you do each step matters. How you get the alignment on the full length matters. How you add things in matters. UP was the first technique we developed that I'm going to show you. And this is the picture. So you have all the sequences here. You divide into the sort of full length sequences, and you get an alignment and tree on it. You get the remaining sequences here. What do you do with this alignment and tree? You are going to take that tree and you're going to break it up into subsets. And the subsets are hierarchical. There's the full data sets, then you break into 2 pieces by balanced cut, and then you break each of those into 2 pieces until each subset is maybe size 10. And now on every single subset, you have an induced alignment. And you take each of those induced alignments and you build a hidden Markov model. So this is now a collection of hidden Markov models that represents that initial alignment. And the reason we're using this collection is that it's more accurate than a single HMM. And this is this thing we keep seeing, that when you have heterogeneity in your data set, a single HMM is not a great solution. It's not a great model. So when you have heterogeneity, you want to break it up into a collection. This collection of HMMs is then used to add the remaining sequences. So what do the remaining sequences do? They each pick one of the HMMs according to the bit score. And then they are added into that subset alignment. And by transitivity, you can say how they get added to the full alignment. That's UP. Now, on 1 million sequences, it's better than pasta. So you get better alignments, and you can run it on 1 million sequences. The real benefit for this approach is that for alignments and trees, it's more robust to fragmentation. So if you have a lot of short sequences, it can actually manage to maintain alignment accuracy, and the tree error also doesn't go up by too much. So it's really, the point is what happens when you have sequence length heterogeneity. That's where you get the benefit. So what's being shown here is the alignment error of pasta versus up. And you can see that pasta has more error than up. And that's a function of how much of the data are fragmentary. Tree error we're showing here is the increase in the tree error, not the absolute error, but the increase compared to having everything be full length. And you can see that there's a small increase for up, but a huge increase for pasta. Now, I'm giving comparison to pasta. Other alignment methods have the same problem. Okay? So you have this problem that alignment methods that are not specifically designed to handle sequence length heterogeneity don't do well. That's the main point. And this technique of taking a data set, dividing it into two, and building a model on the full length alignment, that's the key. Okay, there are other methods that have that technique, right? Do a model on the full length alignment. They differ about how they either build that model or how they use it. So which is basically combining information not from just the one best HMM, but from the top like 10 HMMs, and then combining that information to do a better alignment. And so which is actually improves upon up. HM merge is actually building an HMM, a weird HMM, no longer a profile HMM, to model this collection. And then Emma completely avoids the use of HMMs. So what is Emma doing? It's saying that Math has this really nice technique for adding sequences. It's actually better for full-length sequences, not short sequences. If what you're trying to add are full-length sequences, Math is very good, but it doesn't do that well with high-diameter data sets. So the combination of divide and conquer with Math that's what Emma is. So these are all techniques for how do you add sequences into an alignment. Short sequences, long sequences, okay? I have several more parts, but if anyone has any questions right here, I'll take one question or two. Okay. Comparison, Emma, Which, and Math this is, again, error, and this is Emma. So for a low rate of evolution and you're adding full-length sequences, actually, this has a high rate of evolution, but the point is you're adding full-length sequences. Emma's better. Emma has an advantage over both of them. And Math add, which is the thing that's really good for full-length sequences, not as good as Emma. Okay, so that's one piece of data. Part 3, statistical alignment. So statistical alignment, you basically, the input is a set of sequences, and you want an alignment and a tree. Okay? Or you may only want the alignment, but I'm going to talk about it in the case of co-estimating the alignment and the tree under a model. So you're going to end up getting a sample of the trees and a sample of the alignments, and you get point estimates so that you can actually use them. Balify by Ben Redlings and Marc Suchard from UCLA is a Bayesian method that does this. The problem with Balify is just that it's really computationally intensive. Because it's Bayesian, you're using MCMC, you want it to converge. So you're trying to let it continue until it converges. That can take a long time. And when I mean a long time, it could be months on small data sets. So it's hard. It's hard to use. And they even say, only do it with 50 or fewer, okay? That's really small. Okay, so this is the high-level idea. It's using MCMC to sample. The recommendation is to run it until it converges. You can do that, but it can take a long time. And so what we want to know is how do we make it scale to large data sets? The first question is, can we use it within divide and conquer like pasta, okay, or like up? And we have this paper where we looked at it. And by the way, this is the new stuff that I haven't talked about in previous CGSIs. And so what happens if we use Balify as the base method in pasta? Instead of using Maft, we use Balify. Or what if we use pasta using Balify as the base method within up? Okay? So this scatter plot, it's comparing default pasta to pasta with Balify. And it's a data set with 1,000 sequences. Everything that is this is the column score, which is a way of evaluating alignment accuracy. This is SP score, which is also alignment accuracy. This is recall for homology, so it's getting exactly the full column exactly right. And anything that is above this line is better done by pasta with Balify. So the column score is definitely improved. The SP score, it's mostly improved. That's the recall. This is tree error. So once you get the alignment and then you look at the tree, which is doing better, most of the time it's pasta with Balify. So you can put Balify in on subsets instead of using it on the full data set, and it will improve default pasta. So this is basically saying Balify instead of MAFT for the subsets, and you get an improvement. Okay? But you can also think of this as a way of saying, I can run Balify in a large data set, I just have to do these subsets. Okay, this is the same thing, but now with UP. UP is doing this two-phase thing where some of the sequences are held out. You get a model on the full-length sequences. And here we're saying, suppose the model is, instead of it being pasta, it's pasta with Balify. Okay? And again, everything that's above this line, it's better to do pasta Balify as the backbone, and it's generally above, generally above, generally above, not 100% above. So we can make these statistical methods that don't scale, make them scale by using them on subsets. That's the key idea. And it's not limited to Balify, it's not limited to MAFT, it's really anything. Okay, so far the observations are Balify within pasta and then subsequently within UP, improves scalability and accuracy. Scalability improvement compared to Balify, accuracy compared to using MAFT. Okay? Now, the next thing we did about Balify is that it's co-estimating alignments in trees. It takes a huge amount of time for it to converge. Can we make it converge faster by just saying, we're going to figure out the tree first, and now you just do the alignment? And what if we don't make it converge? What if we just stop? Use what we get. So what's the impact on alignment accuracy of computing a tree in advance? And what's the impact of not waiting for it to converge? So first of all, giving Balify an estimated guide tree compared to letting Balify just run, giving it an estimated guide tree has the lowest error. Here's what it gets if you don't give it an estimated guide tree, and we're just looking at alignment error. And this is MAFT. So this Balify plus an estimated guide tree gives better alignments than MAFT, and they both give better than Balify run by itself. So you're getting better alignments by fixing one of these things, not having a co-estimate. Now, what about two different data sets? Now we're looking at PRANK. PRANK is another statistical method for alignment. We're saying, suppose we give PRANK an estimated guide tree. It's supposed to be very much like Balify. Guess what? It's worse. So Balify really is good. Balify really is good, but you should give it an estimated guide tree instead of trying to get it to do its work by itself. What about conversions? We looked at the ESS values. They were all bad. You know, even if we gave it 500 hours, they were bad, even when they're small data sets. So we have this problem that if you wait for convergence, you may never get anywhere. Maybe you don't need to wait for convergence. You can still get good results. I know that's a bit blasphemous, but I think it's worth considering if you want to use these methods. Okay, so comments right now. Balify is computationally intensive, but you can trick it into scaling. And the tricks are, like, use it on subsets instead of on the full thing. Don't wait for it to converge. Give it an estimated guide tree. These are some of the tricks, okay? Part 4, debates about simulated versus biological data sets. So what is the debate? With simulated data sets, you know truth, but your simulations will never be completely realistic, and so you may really prefer to use biological data sets. The problem with biological data sets is that, in fact, it's not clear that you can rely upon the quote-unquote benchmarks. There's a lot of debate in the literature about how reliable those quote-unquote benchmarks are. And you could read up on it, but some people say they're adequately reliable. Some people say they're really not adequately reliable. So that's the debate. You know, you get realism, but maybe the alignments that are supposed to be reliable are not reliable. So we did a study where we looked at this on protein data sets. only small ones. The largest data set we looked at had 27 sequences. And we let every Bellify analysis run for like 3 months. So we're really giving it a lot of time so that it could converge. And we're looking at different methods and how accurate they are. And we also did simulations based upon these data sets that were also small, 27. So we had 1,192 biological benchmark data sets and 120 simulated data sets. And this was published in Systematic Biology. This is the simulation first. So I'm showing SP score versus modeler. SP score is essentially recall. Modeler is precision. So these are essentially precision recall subfigures. And there are 6 model conditions that vary in terms of the rate of evolution. The top thing is high rate. This is a low rate of substitution and indel. Indel rates here, low, medium, high. So substitution rates versus indel rates. So the easiest case is when everything is low, which is here. Now, Bellify is in red. It's being compared to Clustal, Contraline, MAFT, Muscle, Prank, Prime, ProAlign, and PropCons. Very, very, very good protein alignment methods. Bellify is in red. It's always the best, okay? That's the main thing. Not what's bad, just Bellify is the best, yes? Everyone says Bellify is best? Yes? Okay. Biological data sets changes. So sorry to say we swapped the precision recall, but nevertheless, 4 different collections of benchmark data sets. This is Bellabase, Humstrat, Sisyphus, and MatBench. Here's Bellify, here's Bellify, here's Bellify, here's Bellify. So what is Bellify good for? It tends to be good for the modeler score. So what that means is that if it has 2 things in an alignment, it's probably right in a site. But it's failing to recover a lot. So it's not great for SP score. It has poor recall, has good precision, poor recall. And, you know, this is just really weird. So Bellify is the best on simulated data, and it's in the middle, really, on the protein benchmarks. It's good for modeler score, okay? It's not great for SP score. So few false positive homologies, a lot of false negatives. And the question is why? One way of seeing that is that Bellify is under-aligning. It's basically making longer alignments. A lot of these methods make shorter alignments. And Nick Goldman from Cambridge has been talking for years about how alignment methods have been developed to make short alignments because they're prettier. No one likes these long alignments. And he thinks the gappy alignments are correct, and people should just cope with it. But you know, this is an example. Bellify is making long alignments, but maybe they're not supposed to be so long. Maybe they are supposed to be so long. So what's going on here? It's not because of failure to converge. This is really working okay. So it could be, and here are my 3 hypotheses, and I'd like to know what people think. Model misspecification. So model misspecification is basically Bellify is co-estimating alignments in trees under a model that looks a lot like GTR, but with indels. So, you know, it's the sites are independent, the whatever, 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 it's all, there's no selection, right? Rate variation, sort of normal. Maybe the model's wrong. When you look at biological data, maybe it's just the wrong model. The second one is structural alignments, which is what those benchmarks are based on, and evolutionary alignments may not be the same. And the third one is that the structural alignments themselves are not correct. And there are a lot of people who have said the structural alignments are a problem. I have gone and asked people, what do you think? And what do you think? And some people say, like maybe half of the people say the structural alignments are wrong. Half of the people say model misspecification is wrong. Occasionally, someone will say structural alignments are not evolutionary. And of course, Ben has his own hypothesis, which he proposed a year ago or 2 years ago, which I still don't have an answer to. So, you know, I don't know, but I have to tell you, what scares me the most is that it might be number 1. Because if it's number one, then everything that we're doing in phylogeny is based upon models that don't make sense. So I'm really hoping that it's not number one being the main problem. I hope it's number three, but it could be number one. And if it's number one, we really need to fix the models to make them more realistic, which brings me to, so far people only want identifiable models. They want models they can prove theorems about. Is John Novembre here? No. Okay, well, we had this conversation earlier. Models, if you only want to work with models that are identifiable, you can prove nice theorems, but maybe you're not going to be dealing with good reality. So this is my problem. Anyone have a comment about what they think it should be? The answer?
I have a question.
Yeah.
Maybe a suggestion. That if it's structural alignments that are not correct, and the alignment tools potentially then are giving the correct alignments, couldn't this be validated? So that would be suggesting maybe better structural alignments, right? The computational alignment, sequence alignment.
I see what you're saying. Yeah, you might get better structural alignments by looking at these estimated alignments.
And can that be checked? Like it's not just.
But the thing is, how do you know if something's correct structurally? So the way these structural alignments are being done is typically you're getting some parts of it, and then you're interpolating. So maybe where it's wrong is because of the interpolation. But the other thing is a lot of these methods these structural benchmarks have been based upon not looking at things and making sure everything's correct, but using methods that people have developed that they've trained on these things. So they've trained them on things that aren't necessarily accurate. I mean, there's a literature here. I mean, I think your point is an interesting one, but how would you know? How would you know if it's the correct structural alignment?
Yeah, my point is just that you have now kind of two hypotheses, right? Whether the original structural alignments are right or the ones that are more in line with the sequence alignment. So that it's an easier problem than just being able to say whether the original ones are wrong, right?
Okay, so it's a very good question. And that would be and it's not something I've thought about before, so it's great. So take something where you get a very different estimated alignment from the structural alignment and see if someone can figure out to what extent the predicted structural alignment is actually correct. Okay, good question. So, summary. Progress in alignment estimation has been made. Dealing with large data sets, we know how to deal with large data sets. Dealing with sequence length heterogeneity, we pretty much know how to deal with that. But there's a bunch of questions. How do you assess alignment uncertainty, right? Because if you can use that alignment uncertainty, it could help maybe in downstream analyses. How to use a set of alignments instead of one point estimate? Do you want to keep the set or just use it to get a point estimate? Are there better ensemble methods? T-coffee is an ensemble method, but maybe there are others that we could develop. What are the best ways to merge disjoint alignments? We have one technique. We have now 4 techniques, but maybe there are better ones. And how can we do statistical alignment efficiently and well? Maybe we need to change the model, although I really like your suggestion, which is completely different from all this. Some recommendations for the users. For small enough data sets, MAFT is great as long as you use a good version of it, like MathLincy. There are really good methods if the data sets are small enough and you're working on proteins, like ProMols, Contraline. Balify is a very good statistical alignment method. And by the way, Ben Redling's told me he's modifying the model now to try to deal with some of this. And T-coffee is great for combining things. But for large numbers of sequences, you need to have other techniques. Twilight is a new method that came out that is potentially really good for very large data sets. But if you have sequence length heterogeneity, again, you need to have different techniques for sequence length heterogeneity. And again, genome length, different problem. And summary, multiple sequence alignment, it's hard, but we've made a lot of progress. But we still have things to do, so if people are interested, there's new things that you could do. It's not done. And the acknowledgements, you'll notice, Siavash, he was the developer of Pasta. He was one of the developers of UP. And he can answer like any question that I can't answer. You can just ask him. And then the other people who worked on various steps. Thank you.